Have you ever wished that you could live longer or be able to bulk up without working out? Now, picture a world where malaria is completely eradicated. And now, imagine seeing woolly mammoths roaming the Arctic tundras once again. Sounds like science fiction, right? But it isn't. This is the reality being made possible by gene editing. Now, allow me to take you on a journey through gene editing, from its inception to where we are now, the big questions, and where it might take us next. So, let's start at the very beginning, all the way back in the mid-1800s, when Charles Darwin introduced the theory of natural selection. Now, he didn't know what genes were, but he knew that traits could be passed down through generations, changing species over time. And around the same time, Gregor Mendel, an Austrian monk, was growing pea plants in a monastery garden. Interestingly, he noticed that certain traits followed predictable patterns. Without realizing it, he had uncovered the basic laws of inheritance. While Mendel gave us the logic of inheritance, it was Albert Cossell who gave us the letters, the 4 bases we now call A, T, C, and G. DNA is the instruction manual of life, and genes are like words written in those 4 letters. But we still didn't know what DNA looked like at this point. However, that changed in the 1950s thanks to Rosalind Franklin's photograph 51. That blurry, black and white X-shaped pattern was the clearest evidence yet that DNA formed a helix. And this was the missing puzzle piece that allowed James Watson and Francis Crick to build the iconic double helix model of DNA, the molecule that carries our genetic code. Then, by the 1970s, we weren't just visualizing DNA, we were starting to read it. And fast forward to the early 2000s, the real game changer arrived with the Human Genome Project. It involved scientists from all over the world, took more than a decade, cost nearly $3 billion, and revealed all 3 billion letters of our human genetic code. We had finally read the entire blueprint of human life. But technology didn't stop there. We wanted faster, cheaper, more powerful tools, and we got them. Next-generation sequencing brought down the time and cost it takes to read DNA. What once took years now takes just hours or even days. And then yogurt happened. Yes, yogurt. In 2007, a company named Danisco, which sells microbes and other ingredients that can be used to make foods like yogurt, was facing a problem. Their bacterial cultures kept dying from viral infections, but they noticed that some bacteria survived the attacks. Now, they didn't know why these survivors made it, but when they read their DNA, they found something unusual. These bacteria had acquired a piece of the virus's DNA inserted into their own genome. And that, as it turns out, is the heart of a bacterial adaptive immune system called CRISPR. CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. I know, it's a mouthful, but let me try to explain it to you. So, CRISPR is actually named for the repeating sequences in the bacterial DNA. But what really fascinated scientists weren't the repeats, but rather the mysterious spaces in between them. Because these spaces aren't bacterial. They're in fact DNA from the past viral invaders. So think about that. When a virus attacks a bacterium, and if that bacterium survives, it literally steals a copy of that virus's DNA and inserts it into its own genome. And then later, if that same virus returns, the bacterium uses that fragment to recognize the virus and destroy it. So here's how it works. The bacterium makes a copy of the viral DNA spacer called an RNA guide. And this is kind of like a wanted poster. And it hands this to an incredible protein called Cas9. Cas9 then patrols the cell, kind of like a bodyguard, carrying the RNA guide. And when it finds an intruder with the exact match, It cuts that virus's DNA, neutralizing the threat. This was the elegant natural system that scientists Jennifer Doudna and Emmanuel Charpentier studied and repurposed in 2012, a breakthrough that earned them the Nobel Prize in Chemistry in 2020. They discovered that by programming the Cas9 protein with a synthetic RNA guide, you could direct it to cut literally any DNA sequence that you wanted, and this was revolutionary. Suddenly, CRISPR-Cas9 wasn't just a bacterial defense mechanism, it was a pair of molecular scissors allowing us to cut and edit DNA with unprecedented speed and precision. So, once CRISPR makes the cut, what happens next? Well, the cell's natural repair systems take over. So the most common method, called non-homologous end joining, simply joins the broken ends back together. But it's not perfect. Sometimes it can introduce small insertions or deletions that can stop a gene from working. But there's also a more precise pathway called homology-directed repair. Here, scientists provide a healthy version of the gene containing similar sequences, or homology, which the cell can use to replace the faulty DNA. So this is kind of like fixing a typo in a Word document with the find and replace function. So with this tool, we're no longer just decoding life, we're rewriting it precisely, powerfully, and with implications that are as exciting as profound. So let's start with medicine. For decades, medicine has focused on treating the symptoms rather than the causes of disease, often by targeting proteins, which are the final product of our biological chain. But now, with new technologies like CRISPR, we're moving upstream, directly targeting the instructions that guide life itself, our DNA. In 2023, we witnessed the first ever approval of a CRISPR-based medicine called Casgevy, a cure for sickle cell disease and transfusion-dependent beta thalassemia. Now, as a medical student in Sri Lanka, I've seen how devastating thalassemia can be. Every month, many families travel long distances, sometimes across the country, for their children to receive blood transfusions, and they often miss days of work and school. Until now, the only potential cure available was a stem cell transplant. However, this is only an option available to a small fraction of patients with a matching sibling donor. But now, with gene editing therapies like Casgevy, there's a possibility of a cure without the need for a donor, a future where hope isn't just reserved for a lucky few, but extended to every family. And this isn't just about one disease. In fact, there are over 6,000 known genetic disorders worldwide, and most of them are rare, under-researched, and with no approved treatment. But now, thanks to CRISPR, that's changing. From curing genetic blindness to reprogramming a patient's own immune cells to fight cancer, gene editing is pushing the boundaries of what medicine can do. It's also being explored for diseases like Alzheimer's or muscular dystrophy, and even conditions caused by many genes like heart disease or diabetes. One huge milestone in gene editing is the story of baby KJ. Baby KJ was born with a rare fatal genetic condition where his body couldn't break down the protein in food. And without constant hospital care and medication, toxic levels of ammonia would build up in his body. But a team of scientists and physicians did something extraordinary. In just 6 months, they developed a personalized CRISPR therapy designed just for him. And since then, his condition has stabilized, and as you can see, he's a happy baby who finally got to go home this month. So that was the first ever CRISPR therapy designed for a single patient. And it's a glimpse into a future where even the rarest of diseases can be treated, and no patient is left behind. CRISPR is revolutionizing transplant medicine too. Take xenotransplantation, which is using animal organs for human transplants. Now, normally, this would lead to rejection of the foreign organ by our human body. But CRISPR lets us humanize these organs by removing viral DNA and markers to make them more compatible. And beyond treating disease, CRISPR is also helping us understand it better. In research, it allows scientists to turn genes on or off to observe their effects and even to find the root causes of diseases. But gene editing isn't just transforming medicine, it's transforming the world. In agriculture, it can help us grow crops that can withstand pests, diseases, droughts, herbicides, and even have better commercial properties like longer shelf life, improved appearance, and flavor. And gene editing can also change livestock with applications like improved disease resistance, increased muscle mass, and even removing allergens in products like dairy and eggs. Researchers are also looking into various ways to use gene editing to combat climate change, which is one of the greatest existential challenges we face today. Some solutions being explored are growing crops that can adapt to harsher climates, and even altering the microbiomes inside animal guts to reduce greenhouse gas emissions like methane. And finally, CRISPR may help us protect endangered animals or even bring back extinct ones. As you can see, CRISPR is no longer just a medical tool, but as we extend this power beyond the lab and into the wild, we enter uncharted ethical territory. So, let's start with safety and efficacy. CRISPR may be precise, but it's not perfect. Sometimes it can make off-target edits, changing parts of the genome that it wasn't supposed to. And even when it cuts in the right place, unintended effects can still occur, which may not appear for a long time. There are also major scientific limitations. Our knowledge of the functional significance of genes is still incomplete. Many people carry genetic variants which we don't know the effects of. These are called variants of unknown significance, and editing these genes without knowing their function can be dangerous. Then, there's the issue of representation and equity. Majority of genomic data is based on European and North American populations. Other populations remain largely underrepresented, which can lead to bias in diagnostics and treatments. And when therapies can cost over $2 million per patient, the question becomes, who gets access and who gets left behind? So while they could provide a benefit for those who can afford it, the disparities between health and wealth that already exist across nations could be intensified. Then there's the debate between somatic editing and germline editing. In somatic editing, you only affect the individual patient, but germline editing alters the DNA of embryos and reproductive cells, meaning these changes are heritable and passed down to future generations. So as you can see, the stakes are much higher here because you're not just changing one person, you're changing the entire future of a family line without their consent. There is broad international consensus that germline editing is unsafe and unethical at this stage. Yet, in 2018, it happened. A Chinese scientist announced the birth of twin girls resulting from embryos that he had edited using gene editing to make them resistant to HIV. But this procedure was poorly regulated, unnecessary, and unethical. The scientific community was outraged, and his actions were condemned. However, this served as a global wake-up call on the dangers of unchecked experimentation. And this brings us to one of the most uncomfortable questions about gene editing. Where do we draw the line between healing and enhancement? CRISPR was designed to treat disease. But what happens if it's used to make our children taller, smarter, or more athletic? Are we trying to cure disease or create better humans? This opens, imagine selecting your baby's features, like choosing your avatar in a video game. This opens doors to designer babies and unrealistic ideals. And history warns us of where this path can lead. The term eugenics once justified some of the darkest chapters in history. In the early 20th century, thousands were forcibly sterilized in the United States based on race, poverty, disability, and other discriminatory factors. And these same ideas later laid the foundation for the genocide by Nazi Germany during World War II, where millions were murdered or sterilized in the name of so-called genetic purity. And today, many ethicists and members from marginalized communities believe that that gene editing could be a slippery slope to a new eugenics movement. Similarly, attitudes towards embryo research vary across cultures and religions. While some see it as healing, others see it as playing God. And that's why governance and regulatory oversight are essential. We need guidelines, frameworks, and standards to ensure transparency, safety, consent, and access. Finally, we cannot ignore the potential for misuse. In the wrong hands, CRISPR could be used to developing bioweapons or biological warfare, posing national and global security threats. Now, I'd like to introduce you to one final concept. It's one of the most powerful and controversial developments in this space, which is something called a gene drive. A gene drive is a genetic engineering technique designed to quickly spread a desired trait to all members of a population. They're most effective in sexually reproducing organisms with short lifespans. Take mosquitoes, for example. Don't be fooled by how tiny they are. They are the deadliest animal on the planet, responsible for close to 1 million deaths every year. Not only malaria, they can transmit many diseases like dengue or chikungunya, which are in fact very prevalent in Sri Lanka these days. With gene drives, we can engineer mosquitoes to prevent them transmitting disease. And other applications include getting rid of crop pests or even invasive species that are threatening native wildlife. Sounds incredible, right? Yes, it does, until you remember that mosquitoes are part of a food chain. Our ecosystems are deeply interconnected. Changing one species can have ripple effects across entire ecosystems, disrupting food chains and reducing biodiversity. And mosquitoes don't need passports or visas. They can just cross borders and affect communities without people's consent. That's why community inclusion is so important. Otherwise, we risk solving one problem by creating another. Gene editing is no longer theoretical. It's not 10 years away, it's not 5 years away, it's right here, right now. So, the question is no longer, can we edit our genes? But rather, should we? For whom? Why? How? And who decides? Because the tool itself is not inherently good or evil. It's the user. And decisions this powerful, which affect everyone, cannot be left alone to scientists or policymakers alone to decide, far away from the communities who will actually live with these consequences. We need a global conversation between patients, local communities, cultural leaders, indigenous voices, scientists, ethicists, policymakers, and institutions like the WHO and the UNESCO. Because if we're going to rewrite the future of humanity, it must be transparent and culturally sensitive. So, let's write a story where we don't just change our DNA, but redefine global health and make sure it's a story that includes all of us. Thank you for listening to my TEDx talk.